Just a minute. Yeah, should be better. Okay. Okay. So, hello and welcome to the last session for this course. So, like I've mentioned in the previous recording, the objective of this particular session, this last session, is sort of to recap everything that we have discussed till now, to summarize the entire course so that we can have a recapitulation of all the key concepts and the key studies that we have discussed in this entire tenure, so that you can take away the key findings, you can implement them whenever you wish to pursue, or if you do wish to pursue a research career in the future. Um, Sort of comprehensively mold together the different concepts that we have learned. I am sure that a lot of the concepts were very new. Some you might have come across in your bachelor's or your master's, or somewhere you might have come across. Some of it, I assume, are very up and coming technologies, and it's not something you would even possibly won't even find in textbooks at this moment. So, a better place to study it would probably be in research papers and review papers, recent articles. Find those papers which are very well cited, well reliable, in good journals, and use them for further reference if you wish. And apart from that, of course, I have included as many references as possible. I have referred to a large number, very different, very diverse sources throughout this entire course. I've looked at textbooks somewhere, I've gone through recent publications, I've looked at book chapters, research articles, review papers. It's been a mixed bag. I've used YouTube as a resource as well, because it has excellent animations of certain techniques which um, sometimes you might find a little difficult to visualize, but then people have made really nice schematics to simplify those techniques and make it easy to interpret, easy to understand. So you can take those references as well for your for better understanding. So let's we will brush through, because there's a very large number of slides, 250 slides that we have covered over the course of these classes in 24 classes. So that's a lot to be covered. So I will not go through the details of it, but will summarize everything. Um, some of these topics, of course, I've already summarized, because we have had exams in between, during which time I have recapitulated some of these topics for ease of understanding. Nonetheless, our initial discussion was on very core, very central molecular biology, whereby we first had discussed the discovery of DNA, the structure of DNA, how the genome is organized, how is gene function regulated, and these were mentioned as separate topics because it was very early on in the course, and then gradually we have moved forward towards more condensed topics, which is why you have not found a lot of bullets on it, but a lot of topics have been covered nonetheless. So initially, we understood how DNA structure and how the role of DNA as the master molecule in the cell was unraveled by some very um, key landmark experiments which were carried out by these stalwarts: Frederick Miescher, Oswald Avery, Maclean McCarthy, Frederick Griffith, and the initial biochemical investigation of DNA, which was carried out, whereby the fact that DNA stores genetic information was understood when people inoculated Streptococcus virulent strain uh, virulent strain injected into mice caused the death of mice. However, when live, non-encapsulated, non-virulent strain was injected, then mice did not die. And when a mixture was made of live bacteria with the heat-killed virulent bacteria, then the mice were living. And when heat-killed virulent bacteria were added to non-encapsulated, let me use my cursor, non-encapsulated, non-virulent bacteria, and this mixture was made and then injected into mice, then the mice were not able to survive. So this is how Frederick Griffith had shown earlier that adding this heat-killed virulent bacteria to a live non-virulent strain permanently transformed this second strain into a lethal virulent encapsulated form. So it was experiments like these, which in today's time you might find very surprising, but these were landmark experiments which established the foundation of biology as we know it. Then came Avery et al. As well, who concluded that DNA extracted from the virulent strain actually carried the message for it to be deemed or declared as virulent. Uh, and later on, the Hershey Chase experiment, yet another incredible experiment, which showed that, which provided independent evidence. Of course, I've not shown it as a timeline, but these were simultaneous research groups who were working on kind of similar concepts, trying to understand a similar problem. But their findings were quite independent of each other and indicated the same thing. So they provided independent evidence that DNA actually carried the genetic information. So they took bacteriophage cultures, which were separately labeled, one of which was labeled with radioactive isotope of phosphorus, which labeled the DNA. The other one was radio labeled with sulfur, which labeled the protein code. And then both were independently allowed to infect bacterial cells. Centrifugation was performed, and then cells were checked to see whether or not you find it in the pellet, whether or not you detect radioactivity in the pellet or in the supernatant. And of course, the fact that bacteriophage DNA is what is the actual infective um, component of the virus renders phosphorus as the material which will get passaged or inherited by virtue of that infection. And so the infective characteristic or the genetic information for for that virus is carried within the DNA. That could be clearly concluded at this point. Then came Francis Crick and James Watson, who collaborated, who showed the DNA structure. Of course, we must definitely mention Rosalind Franklin here. We have previously mentioned her as well. So Linus Pauling and Maurice Wilkins were also actively working in this field. In Wilkins' lab, Rosalind Franklin was the one who was extensively working on X-ray diffraction methods, and she was able. Her results later analyzed by Watson Crick and also eventually Maurice Wilkin led to um, them being awarded the Nobel Prize in Medicine in 1962. So this is what DNA pretty much looks like. You have a phosphodiester backbone, you have nucleotide bases, which have three phosphate moieties attached to them as well. And PPI gets released, or inorganic pyrophosphate gets released, and one single phosphate gets incorporated into the backbone, forming the continuous backbone chain that we are all too familiar with. And apart from that, you also can see how the templated polymerization works, which is what I just mentioned. And finally, you get this double-stranded DNA. This double strand is not linear. It is organized into a helix. And the DNA double helix is what we have learned about extensively in all our priming years. So the structural integrity of the DNA double helix is largely attributed to some key findings which were made by Shargov as well. So Shargov's rules is how we commonly know it, and there are a couple of rules. Basically boils down to 3 or 4 facts. First, that base composition is very species specific. Among different species, you'll find a lot of variation. Second, for a species, however, this composition or this abundance does not change a lot with the age of the organism or the nutritional state or the changing environment. And the main finding, which is usually associated with Shargov's rules, is the fact that A plus T equals G plus C. And basically, the balance between purines and pyrimidines is what he has talked about. And these have actually laid the foundational stone in establishing the 3D structure of DNA eventually, and how genetic information is encoded within the DNA, how it is read, how DNA is opened up. All those findings came much later, but these were foundational studies. Then in 1953 came Watson and Crick's 3D model of DNA. So that's how it basically looks like. Again, think of the journey we've had from understanding the structure of DNA, the biophysics of DNA, and from there to using DNA as a biomarker. It's been a beautiful journey, I must acknowledge it. So there are two helical chains in the DNA double helix, which are wound around each other, most commonly in a right-handed fashion. There are three types of DNA as well. That's not something that we discussed, I think, during this course, but if you're interested in it, you can read about it from Albert Leniger, A, B, and Z form of DNA. And they are present in nature and they are present at very unique locations. You can, it's an interesting thing, but it's not really the idea behind this course. Then the hydrophilic backbones of alternating deoxyribose and phosphate groups are on the outside of the double helix, facing the aqueous medium. The furanose ring of each deoxyribose is in the C2 prime endo conformation, and the purine and pyrimidine bases of both the strands are stacked inside the double helix with their hydrophobic and nearly planar ring structures very close and perpendicular to the long axis. Um, the pairing of the two strands creates a major groove and a minor groove on the surface of the DNA double helix or the DNA duplex. So there's a major groove, and that's where the transcription factor sits pretty much here. We have discussed it later as well. And there's a minor groove also. Now, the definition of a gene is the unit of hereditary information that occupies a very fixed position on the chromosome. So, a definite stretch of coordinates are usually annotated as a gene because of the composition that it has, because of certain um, cis elements that it is usually surrounded by, which demarcates it as a gene. And yeah, so this is what chromatin pretty much looks like. It's a double helical form of DNA there is extensive packaging which it goes through. Maybe I'll come to it later when we discuss the epigenetics part. But basically, there are nucleosomes based on a string structure, then that forms a chromatosome, then that forms the chromatosomes. They line up in this solenoidal sort of fashion, then they form the 250 nanometer fiber, then finally that undergoes extensive coiling to finally form the chromatid of a particular chromosome. So it's manifold coiling which finally leads to the formation of a chromosome. Um, so these are the major components of the human genome. So you have protein coding genes. Out of these form a very tiny percentage, only 1.5%. But apart from that, you have lines, signs, retrotransposons, single sequence repeats, segmental duplications, miscellaneous heterochromatin, miscellaneous unique sequences, etc., which all together form the entire human genome. And these elements, they all have different functions as well. Again, coming back to the discussion we were having today about, in having not today, in the previous class about cytogenetic biomarkers. So this is the human chromosome set that I talked about, that gems are stain and also artificial coloring, both of which they represent the karyotype of the complete set of human chromosomes. And when there are abnormalities, then that includes some sort of reciprocal translocation or some sort of alteration, then that can sometimes lead to changes. Um, and that can actually be used as a biomarker as well for certain diseases that includes AML and other hematological malignancies as also. Then you have the central dogma of molecular biology, from DNA to mRNA to protein. And when you define a gene structure, which is what I was previously mentioning, that there are certain cis elements which define the boundaries of a gene or the foundation of a gene, then that includes certain regions like, of course, the ORF, the open reading frame, which includes intronic and exonic sequences, which have some downstream regulators like terminators, enhancers, silencers, etc. There are some upstream regulatory sequences, which include distal control elements and proximal control elements as well. And sorry, yeah. So information is passed from DNA to RNA to protein as part of the central dogma. Then, yeah, this is what it looks like, and how much information is condensed into the chromosome was reflected by this particular schematic, where you can see that in chromosome number 22, you can see that around 48 to 20 power 6 nucleotide pairs are present. And from here, if you look only at 10% of the chromosome arm, that itself has 40 genes. From there, 1% only has 4 genes, and in one gene, you have 3.4 into 10 to the power 4, or around 3,400 base pairs that are present. So that's 34,000 base pairs that are present. That's a very big number. Getting it wrong. No, it's fine. 3. Then you have different chromatin architecture also having a role to play. Like, for example, here you can see a chromatin loop. We have discussed this extensively when we talked about high C, 3C, high chip, and all these chromatin chromatin looping and chromatin architecture associated techniques. We have discussed it towards the fag end of the course. I'm sure it must be quite fresh in your minds, but yeah, so you have certain promoter elements, you have enhancer elements, and you can see that for each of them, the type of modification on the histones are quite variable. Like, for example, H3K4ME3, H3K27 acetylation versus H3K4ME3. Instead of H3K4ME3, you have Me1 along with H3K27 acetylation that dictates something else entirely. And uh, depending on what sort of transcriptional control is installed, some polymerases can actually be more efficient and can generate multiple transcripts, or rather, you can also interpret this as an abortive transcription measure also. So there can be an imbalance in different polymerases proceeding differently. Then you have CTCF like elements bringing together different domains of different chromosomes to form TADs, and that has an incredible role. Apart from that, regulating gene function occurs at multiple steps, where from DNA to RNA, RNA to protein, you have these many steps. Whether or not RNA will be synthesized, whether it will be translated, whether it will be, and it will undergo the processing steps following translation, all of that. These are different steps at which gene function may be controlled. And like I said, the 3D chromatin architecture makes a lot of difference. Like, for example, linearly very distant locations do come across or do actually directly interact with each other under certain circumstances. Of course, that involves a number of other proteins, which are usually sort of scaffold proteins that are present there and which bring together really far off domains. So, this is the gene control region or GCR for typical eukaryotic gene, where you have a promoter where the general transcription factors and the polymerase assemble. You have cis sequences, which are binding sites for transcriptional regulators, which can be located adjacent to the promoter or upstream of it, or even within introns or entirely downstream also. So all of these locations can be brought close to each other, and there are multiple proteins which actually facilitate the formation of these 3D structures. Um, Then we have different repressor proteins also doing their job. So repressor proteins can work in multiple ways. Either they can competitively bind to the same site as the activator and prevent the transcription of the gene. They can mask the surface to which other downstream activators are able to bind. Then you can have direct interaction with other general transcription factors and preventing transcription from proceeding, like you can see here. Sometimes it can recruit chromatin remodelers, which will wrap up or remodel nucleosomes and not allow transcription to proceed. You can have the recruitment of histone deacetylases, um, which will remove the acetylation mark and will promote the wrapping up of the chromatin. You can have recruitment of methyl transferases, which will increase methylation and again do the same job. So that's how repressors sometimes can work. Then you have insulators as well. For example, this is an insulator which brings together these two insulated elements, and sometimes that can control that only one gene might be expressed at one point of time and the other is not allowed to proceed because of a barrier sequence being present. There can be multiple rounds of regulation. Then you can see that multiple inputs can actually finally dictate what decision can be made at a promoter. So you can have a weakly activating assembly, you can have a strong inhibitor, you can have a strongly activating assembly, you can have a neutral assembly. The some integration of all of these signals is finally interpreted by the cell to make the final decision. So that was our first class. Then we moved forward and we moved towards methylation and mutation and subtypes. So DNA methylation, like we have discussed again during our last class on epigenomic biomarkers, is an incredible arena that is currently a very hot topic for study. It's naturally an epigenetic modification of DNA. It's necessary for a whole number of processes, and DNA methylation patterns show a lot of variability in context of cancer, like there can be genome-wide hypomethylation that overall the methylation mark is reducing, but for certain specific genes, hypermethylation might occur, that there is excess methylation at certain sites, which sometimes can also occur simultaneously in the same cell. Now, the loss of normal imprinting contributes to several inherited genetic diseases also. That includes Beckwith-Wiedemann, Prader-Willi syndrome, Angelman syndrome, etc. And this is what cytosine versus methylated cytosine look like. You can see there's a methylation at the 5' position in the nitrogen base. Then you can see that hypermethylated pericentromeric heterochromatin, and near the TSG or the tumor suppressor gene, tumor suppressor gene, there can be CpG islands which may be present, which if hypomethylated can lead to loss of TSG expression, a loss of tumor suppressor gene expression. When that is combined with mitotic recombination, genomic instability, it can altogether lead to cancer. So again, it's not as simplistic as I'm trying to convey right now. It's a very fine balance that's maintained in this context. So that again needs more in-depth study. So again, this pretty much shows how DNA methylation is disrupted in cancer cells. I'm going to move past that. Then we'll talk about mutations. We talked about all of the different types. We talked about the fact that they can be spontaneous or they can be induced mutations, spontaneous, which come by virtue of the DNA polymerase machinery not working properly, versus when we talk about induced mutations, usually that involves some sort of a stimulus which has um, affected the sanctity of the bases that are present there, and it has changed the base pairing capability of the altered nucleotide, and it has induced a mutation in response to some sort of a stimulus. And in context of mutations, a very important concept is of the genetic code and all of its different attributes. That includes the fact that bases are degenerate, they're non-overlapping, the genetic code is commaless, non-ambiguous and universal, with a few exceptions, but largely it is universal and non-ambiguous as well. When we talked about all of the different types, we talked about point mutations, which are changes in a single nucleotide base in the DNA sequence. That includes silent mutations, missense, and nonsense mutations. Silent is one which does not change the amino acid. Missense, which completely changes the amino acid. Nonsense is something which changes the amino acid to something that terminates translation at that point. So that's a nonsense mutation. Then you have this normal protein coding gene versus mutated gene and the difference between them. Then we talked about frame shift mutations, which literally, as the name suggests, leads to a shift in the reading frame of the ribosome. So indels of nucleotides may result in a shift of the reading frame or insertion of a stop codon. So either there can be a single base shift, which is going to lead to a complete change in the amino acid sequence, or it can be in frame, which means that if there are three bases that are getting inserted, the resultant outcome is that the reading frame has not shifted, but an amino acid has either gotten added or has gotten lost. So potentially that can affect protein function. So that's an in-frame mutation. So this is sort of a summary of all of these different mutations that I just talked about. Then comes the question, how do mutations arise? So when we talked about spontaneous mutations, I mentioned the fact that there can be uh, DNA polymerase machinery errors or DNA replication errors that form a major class in this context. So that leads to very random, very spontaneous and uncontrollable mutations in the genome. And chemically, another thing that happens is that uh, bases can sometimes shift between different states, which is often also understood as tautomerism. So there are two states in which bases can exist, which is a keto form and an enol form. So between normal pyrimidines and normal purines, there is a certain type of interaction. So for purines, however, they do undergo this tautomerism, and they can lead to some um, unusual pairings. So, for example, while AT and GC would normally pair, in their rare forms, these purines can form AC and GT base pairing as well, which is a non-Watson-Crick form of base pairing, and that if persisted, if that is something that gets duplicated um, or uh, reflected in the second strand of DNA as well, then that can be quite a bit of a problem. That's going to eventually become a mutation. Spontaneous chemical changes involve two types of changes, depurination and deamination. Depurination literally means the loss of a purine, leading to the formation of a deep epurinic site. And deamination literally means the removal of an amino group or an amine group and an H2 group. And there can also be formation of dimers, pyrimidine dimers. Specifically, it's majorly found for thymine dimers, like you can see here, TT dimers, and these are really large obstructions for the movement of polymerases at the time of replication, and it does lead to the formation of these um, stalled replication forks from where replication is not proceeding. It is unnecessarily hijacking the machinery, and eventually either the cell has to repair that, which can be done by NER, which is nucleotide excision repair, or alternatively, that machinery has to be um, sort of removed or stripped off from that replication fork. So that's what happens there. Then you have chemical mutagens, which includes base analogs. Now, again, not going to repeat it, but basically, what happens there is agents like 5-bromo uridine. What it can sometimes do is it can get intercalated or incorporated into the DNA sequence in the normal state. However, if incorporated in the rare state, then sometimes it can lead to changes whereby upon DNA replication, it can undergo transition mutations, and it can cause a mutation in the DNA base pairing because it also undergoes these changes. Then there is mutation by addition. Some intercalating agents like proflavin, acridin, and ETBR insert themselves into Hs and bases, causing the helix to relax, and it can also sometimes lead to mutation. And that can cause single base or multi-base changes also. Next, in week 3, we discussed some different topics. We talked about polymorphism. Generally speaking, any genetic polymorphism is defined as a change or a variation in the DNA sequence that occurs at least in 1% of the population. That includes SNPs, microsatellites, small insertion deletions, and SNPs, of course, are single base pair or single sequence alterations where one nucleotide is substituted for another. So that can sometimes affect the encoded protein. Most of the time, it is not really effective. And you can see that types of SNPs include coding SNPs, silent coding SNPs, non-coding, and linked SNPs. Coding SNPs, if present in the coding region, they alter the protein function, changes amino acid sequence, and that is coding in nature. Non-coding usually occurs in the regulatory region, usually promoter or upstream regions, which might or might not affect the amount of protein produced. Then you have linked SNPs, which are outside of the gene body and they do not affect the function of the encoded protein, but they are SNPs which are somewhere close. Then you have silent coding SNPs, which are present in the coding region, but they do not alter the protein's amino acid sequence. So they are present, but they are not really getting translated into a problem. So this is mutation versus polymorphism, how there's permanent alteration, there's presence of more than one allele. It's a physical versus population attribute, and a number of different differences. Based on genetic polymorphisms, There can be a difference in the way in which an individual responds, like there can be different pharmacokinetic and pharmacodynamic changes. Receptors, ion channels, enzymes, immune system. For pharmacodynamic, pharmacokinetic includes absorption, distribution, metabolism, and excretion as well. There can be alterations in all of this. This concept I've already explained in our last pre-exam class, so I'll skip it. Um, but these SNPs that are present sometimes can indeed change the protein conformation and function via gene or genetic linkage. Um, Intronal SNPs can also influence gene expression through these regulatory elements, and intronal SNPs influence protein synthesis by mRNA splicing and regulation of lncRNA function. In second. When we talk about understanding functional genomics, it's a study of how genes and intergenic regions of the genome they contribute to different biological processes. So, basically, somebody tries to study genes on a genome-wide scale with the hope of narrowing down a list of candidate genes. Again, bringing us back to biomarkers. So, it focuses on the dynamic expression of gene products under a specific context at a specific time, and then it tries to basically integrate um, multiple data sets or multiple results into one common ground for finally understanding what the cell is doing at a particular point of time, like I said, at uh, the moment that you took the snapshot. There are different sort of tools which are used in this context. You can go for survival based strategies. You can use facts based strategies to sort out those or to screen for those cells which are representing the exact model you wish to study. You can go for an arrayed approach, or you may go for single cell functional genomics. So these are all different alternatives for the same. When we talked about sequencing methods, we looked at first generation sequencing. Yeah, but before that, in 2003, the entire human genome had been sequenced. Its idea was basically to look at all of the genes that make up the entire collection of genetic material. In the first generation method, two strategies were used. The first is Maxim Gilbert and Sanger chain termination method. We have discussed that extensively. The details of it you can look at when you want. In today's time, Sanger sequencing method is what we practically use in the lab to validate the exact sequence of whichever region it is that we are interested in, and it is um, considered to be fairly accurate. Then we talked about the Sanger method in more detail. Such a simple but extremely interesting concept, whereby the idea is that you basically terminate the process of chain elongation during polymerization, because you use a dideoxy NTP, as a result of which there are due to the lack of an OH group, extension cannot take place. So the lack of the hydroxyl group causes the polymerization procedure to stop, and thus chain elongation is stopped. It cannot establish a bond with the 5' phosphate of the following DNTP. And Sanger method creates four different reactions. Previously, it used to be differently tagged. Currently, fluorescence tagging can also be done. That gives you very nice, very clean peaks in the chromatogram for each location or each position in the region that you're sequencing, and based on how well your peaks are without any shoulders and more tight sequencing or better sequencing, you'll get these outcomes. Um, Sanger method, now, like I already mentioned, utilizes fluorescently labeled DDNTPs and capillary electrophoresis, and each DDNTP is differently labeled with different fluorescent dyes. So that is what we see here. Then we introduced next generation sequencing, NGS, a massively parallel sequencing procedure that focuses on ultra high throughput, focuses on scalability of your technique and how fast you can go about it. So it's basically used to determine the order of nucleotides in entire genomes or targeted regions of DNA or RNA. You can either sequence entire genomes, sequence entire RNA, or you can figure out different specific conformational regions of the genome. You can do a lot more, which you know much better now based on our discussions in the last couple of classes. But initially, we did discuss the differences between Sanger method and the NGS method. NGS is much more high throughput. It also is prone to more bias compared to Sanger. Of course, if you're working with that large or that extensive a data set, it's bound to give you some false hits. And the strategy by which it works is also very different. We did discuss that also. Um, Vaguely speaking, the entire NGS workflow can be broken down into four steps. First is preparing the sample, preparing the library, and finally analyzing it. So second generation DNA sequencing comes in at this point, you have fragmentation of the DNA, cloning into vectors, which are then transformed. Cycle sequencing is carried out, polymerase generation is carried out, electrophoresis is done, and then you can finally draw whatever conclusions it is that you wish to proceed with. Then we talked about the fact that second generation is used in a number of cyclic array sequencing methods that includes commercial concepts taken forth by Solexa 454 pyrosequencing, the solid platform by Applied Biosystems, the pollinator, the heliscope, etc. So they actually rely, these methods that I just mentioned, they actually rely on emulsion PCR so as to amplify clonal sequencing features. So basically, you generate these little droplets that include both your template, the NTPs, and solid support. And within that, uh, because of the consolidated unit within which this reaction can take place, it's much faster. So a low template concentration, basically proper dilution, results in most bead containing compartments having either zero or one template molecule present, and in productive sort of emulsion compartments where both of these molecules are present, PCR amplicons are captured to the surface of the bead, and then they can be selectively enriched. You can sequence those, so that's a different idea ball game altogether. Excuse me a minute. Yeah. Next, we talked about the Solexa technology, which relies on bridge PCR or the cluster PCR method to amplify clonal sequencing features. So basically, you would have in bridge PCR, you would have this solid support surface or the flow cell, and both the primers are densely present. They're attached by a flexible linker, and the amplification products, they are tethered near the point of origin, and you generate these clusters which all have the same sequence, which can undergo multiple rounds of sequencing, making it even more accurate. Yeah. So, the Illumina method is something you can, of course, refer to based on the video that we had previously discussed. Then we talked about the the science behind the bridge PCR method. Again, this is something I think I had put in the exam questions as well. So, basically, the idea is that you use these chemically modified nucleotides, which are often also called as reversible terminators, because um, it basically a chemically cleavable moiety is present at the 3' position, 3' OH position, that only allows one base pair incorporation to proceed in one cycle. And so, along with it, one of the four fluorescent labels would also be present, which also makes it, which is also chemically cleavable, and that corresponds to the identity of the nucleotide, which has gotten incorporated. Now, after there is one base extension and image has been acquired in all four channels, you will be able to detect that, okay, base A, T, G, or C, which among them has gotten incorporated. And after this extension has been completed, chemical cleavage of both of the groups is done so that now extension can proceed by another base and the whole cycle can be repeated again. That's how it is done. Now, this is being done so fast that it doesn't even feel like such an elaborate process. However, it is a bit elaborate, of course. Then emulsion, then we talked about template amplification once again. So first, if you look here, we just discussed the sequencing strategy, but then we went into template amplification strategies, which basically is a very crucial step so that we can increase the starting material without having to unnecessarily um, isolate that much starting material from cells. You can amplify it using these different methods. The first one, of course, is emulsion PCR, like we had just mentioned a while ago. It is followed by multiple companies, very large, very big shot companies like 454 pyrosequencing Roche, solid sequencing by Thermo Fisher, then Gene Reader by QIAGEN, Ion Torrent again by Thermo Fisher. So they all use these strategies as well, but it involves the formation of an emulsion, then amplification directly on the bead surface, and finally the final product can also be availed from it. Yeah, then we come to solid phase bridge amplification. So that is by Illumina, like we just discussed. There's binding of the template. There is amplification on the surface by bridge amplification. Then generation of clonal clusters, and then within the patterned flow cell, you get increased cluster density also. So yeah, I'm not going to go with the text. You can refer to it on your own time. Then we talked about solid phase template walking also. Very similar to amplification used by Illumina, but there's no uh, the the amplified fragment does not bend over itself. Instead, like you can see here, there is binding of the template, there is primer walking, and there is template regeneration, and then formation of clusters. So it also relies on cluster generation, but the procedure is a little bit different. Then you have in solution DNA nanoball generation, which leads to multiple rounds of rolling circle mode of amplification. So it generates this sort of concatenameric structures, which are called as DNA nanoballs, where intermolecular interactions keep the nanoballs cohesive and separate in solution. And there's iterative ligation by four different adapters and hybridization onto a pattern flow cell, which is then finally used for sequencing. Then this is sequencing by ligation. We discussed this recently, but basically it involves the use of these sort of probes, which have two bases, and you use these N+2 probes, and then no, you use these probes with two known bases, and the rest three are unknown, and you keep repeating this over and over and over again. Then in the next round, you use an N+2 offset so that your already sequenced bases do not undergo the same round of sequencing once again. And you can then fill in the gaps, and you can then figure out what is the complete sequence um, of your target stretch of nucleotides. Then for complete genomics, DNA is sequenced using the combinatorial probe anchor ligation or CPAL approach. So after DNA nanoball has been deposited, you can use, it's very similar to what we just discussed in the previous strategy. So here also you use these single base encoded probe. In the previous one, we knew the sequence of first two bases and then used an offset anchor. Here you have a Yeah, here you have a known base at n+1, and you can use an anchor with n+5, and you can just continue with it. You can read more about this in the reference I've cited below. Again, for Illumina, the reversible terminator logic is used. For gene reader, similar yet chemically different entities are utilized for the sequencing protocol. And again, you can read about it. Here also, imaging is to be carried out. You add the nucleotide, you image, you cleave, remove the modifications, proceed with the next round. That's pretty much how it works. Then we continued this discussion a little bit further. We talked about 454 pyrosequencing by Roche. That is a starkly different strategy, however. It involves these sort of immobilized oligonucleotides onto your non-reactive bead, along with some smaller beads which contain an enzyme known as ATP sulfurylase, along with luciferase, which can be used to detect the readout. So this is the pyrosequencing strategy. So as a base, a single base keeps on getting incorporated, the release of the pyrophosphate, hence giving rise to the name pyrosequencing, is actually detected in an enzyme cascade where it is read as a light readout. And in every single time a nucleotide is added, you will get this different colored readout, and you will be able to figure out what is the next base that is present at that position. Yeah, then comes the ion torrent method, which involves the use of this sort of semiconductor, which monitors how every time an H plus ion is released, there is a sensor which is able to detect it. Then, if we broadly classify sequencing technologies, they are of two types. You have short read and you have long read sequencing technologies, each of which has their own pros and cons. Again, this is something you can refer to from our original class sites. But basically, the idea is that you get um, much more details when you carry out these long read sequencing. That is restricted in your short read sequencing techniques. So they do not span structural variants or other important classes of genomic aberrations, which is actually covered by long read sequencing technologies. It can assemble complete genomes, it can identify mutations, which is lacking in case of short read methods. But it has its own set of advantages also. So you can refer to that. And also, we had discussed this previously as well. Uh, Real-time long-read sequencing method is carried out by Pacific Biosciences, for example. This is the Smart Bell template. If you remember, its unique architecture is what gives it an edge. So you have these double-stranded hybridized regions, and along with that, you have these sort of loops, hairpin sort of loops towards the end, which is where your sequencing primer binds and from where polymerization can actually take place. These are also called ZMW waves, which are zero mode waveguide wells, where sequencing takes place. And all four DNTPs are ready, available for incorporation. And as a nucleotide gets incorporated, light is emitted and gets sort of recorded. The template here is the most unique because it contains these teeny tiny smart bell templates are present, and during incorporation it sort of continues in a it proceeds in a continuous manner, and then it gives you very long reads because there is much more confidence also in this data. Then for synthetic platforms that do the same thing for Illumina, um, so what exactly is done? There is that DNA indeed is fragmented, but then it undergoes barcoding, and after that, those those fragments which are from the same well, they will all carry the same barcode. They can then be pooled and sequenced, and then they can be sort of aligned to get an idea of how exactly these sequences were present. So some continuity is maintained because of the barcode. And for 10x genomics, it also involves something similar. So it simulates the readout that you would get from a real long read sequencing, but instead of spending that much money, it does so artificially by linking these reads close to each other. So here also emulsion PCR, formation of gems, and all the reads from the same gem are derived from the long fragment, therefore they are linked. So then amplification and finally pooling is carried out. This was the sequencing of genome, epigenome, and transcriptome. We'll discuss this in more detail. So I'll move on to the next slide. Um, what is the exome? Exome is the sequence of all the exons in a genome, reflecting the protein coding part of the genome. So WES is what we discussed initially. Again, you can quickly recapitulate the key steps, but you sort of fragment the genomic DNA, you ligate linkers, hybridize it onto a solid support or a flow cell for target enrichment, and then you perform sequencing. So what segregates it from whole genome sequencing? This is whole exome, which gives you readout of only the exonic regions of the genome. Whole genome looks at everything exon, intron, and everything. No selection. All parts are studied. All parts are characterized. It can go only to moderate read depth versus for whole exome sequencing. It gives you variable read depth, has much greater sequencing depth, also more cost effective, but whole genome has a higher price. However, it is better at understanding chromosomal level alterations, and it's much easier, much more accurate at identifying them compared to whole exome. It gives you incomplete information. So whatever you might learn about these aberrations from whole exome sequencing does need further validation. So principally, first step is DNA capture and DNA enrichment, followed by high throughput sequencing, and then comes data analysis. So these are all the steps. In sample preparation, you have to extract DNA, purify it, perform QC. Then you proceed with target enrichment, which is also known as template amplification, whereby DNA is fragmented, not just amplification, but rather DNA is fragmented, exome is captured, washing and elution, etc., is performed multiple times to get rid of background, and then sequencing is performed, followed by data analysis. So again, these were all very technical details, not going into detail of those, but first step is preparing the sample, then capturing and enriching specifically for exons, since we are discussing whole exome sequencing. Common kits have been mentioned here: Agilent, Sure Select, IDT, Illumina, Roche, etc. So based on our requirements and based on standardized protocols, target enrichment for sequencing needs to be performed. Following this, library construction is carried out, which involves repairing the end of enriched exonic DNA fragments, attaching connectors, etc. All of this needs to be done. Um, then comes the sequencing step, which involves, of course, next generation sequencing. This is much more advanced, followed by data analysis, which involves a few more steps also, which again, I'm not going into detail right now, but we'll look at it in a couple of slides. When we talk about exome capture and enrichment, what does that involve? So you use some exome capture kits, which can be commercially purchased also. What they do is first, of course, DNA has to be fragmented, adapter has to be ligated, and you preferentially what your targets. So after shearing and by ultrasonication or biologically by enzymatic digestion, the fragment ends are blunted and then adapter ligated. Now, these use a shearing free transposase based library preparation. Then, targeted enrichment of exonic regions follows library preparation. and methods can actually vary between capture platforms. Um, Agilent's Shaw Select Human All Exon, Nimblegen's SecCap Easy Exome Library, and Illumina's TrueSec, they all use the hybridization method. Again, you can learn about these in your own time. This is a comparison of all of the exome capture kits. You can see that the target site, probe size, number of exons targeted, fragment input, recommended DNA input, the strength, their weaknesses, all of this can be comparatively studied here. When we talk about library preparation steps, we have talked about TrueSeq library. First, you fragment your DNA, you do end repair and select your library size. Then you adenylate the 3' end, where it is added to the end of blunt fragments to prevent random ligation also. Which reduces concatenator template formation. Then you ligate adapters, which involves ligating multiple indexing adapters to the end of DNA fragments, preparing them for hybridization onto a flow cell. Then you can enrich for DNA fragments also. Uh, Yeah. Then you validate your libraries, you hybridize the probe, you capture those hybridized probes using SMB or strep-gravidin magnetic beads. Again, if there's any confusion, please refer to YouTube tutorials or refer to this week's class for better clarification, because right now we're just rushing by, so some information might seem a little crazy. Perform second capture, clean up your captured library. Again, this is something which is done by the company if you are outsourcing it, so it's not your headache at all. But it's a good idea to know these steps just in case you ever need to, or you land up in a situation where this knowledge is might just actually help you. So sample processing, data pre-processing, variant calling and annotation, and finally data utilization. These are the four steps. Then we took a little bit of a detour, talked about epigenetics. We talked about the fact that gene regulation is not just restricted to the gene sequence, but rather the control over the gene sequence or the modification of the gene sequence. So, yeah. So we talked about how epigenetics has a role to play in this context. Proper packaging of the chromatin is survival essential. So we talked about that. Then we had moved on to our discussion on DNA binding proteins. We talked about different types. We talked about the fact that different patterns of DNA cleavage can actually indicate the accessibility of the DNA. Okay. As I say, let's talk about the fact that there are different types of DNA binding proteins, which include histones, which include transcription factors, chromatin remodelers, etc. You can study the type of protein. You can actually use MS to study details about them. When we talk about histones, largely there are four core histones. You would have H2A, H2B, H3, and H4, which altogether form the histone octamer around which your nucleosome bound DNA is actually present. And in our initial discussion, we had discussed that 157 to 160 base pair long oligonucleotides are usually wound around this core histone octamer. Now, beyond this, you have certain linker histones, which usually is histone H1 that actually demarcates a chromatosome, and that chromatosome is actually what defines the end of the nucleosome. Now, if there are no histone H1s, then there is a lot of flexibility. DNA can open up, can close, and that is also known as nucleosomal breathing. That's also a very commonly observed phenomenon. Uh, yeah, so nuclease cutting sites would vary based on how much accessibility is provided by these bound proteins, and this is what the beads on a string structure actually looks like. You have these nucleosome bound DNA molecules present everywhere. They can be separated and they can be studied also to get an idea of the molecular weight of the histones which are present in this DNA molecule. The genome size and gene density information can also be seen here. Then we talked about the nucleosome in a lot of detail. Like I already mentioned, H2, H2, H3, and H4 are the four units that form this nucleosome. And many such nucleosomes in tandem are present, which produce the beads on a string structure that we previously saw. Histone octamer actually is comprised of two dimers. So you have H3, H4 molecules forming one tetramer. You have H2, H2B molecules forming another tetramer, and together they form this octamer. So next comes the fact that whenever epigenetic studies are carried out, enzymatic digestion is quite common. And so that involves digestion with two types of enzymes. One is MNase, which is micrococcal nuclease, the other is DNase. And we get different patterns for each of them. Now, why does that happen? It actually is dictated by how these enzymes are able to sit on the DNA and make the cuts that they do. Like, for example, MNase works like bolt cutters. It straddles the DNA and then cuts it. And therefore, that's what it pretty much looks like. And if you look here, this is your DNase one. DNase one works differently. It attaches to the DNA. It makes cuts in one or the other of the two strands. And when DNA is laid along a surface, DNase one can make these nicks, but only DNA that is farthest from the surface can be easily cut. Histone H1 occupies a position outside this octamer, forms the chromatosome, like I already mentioned. Then the histone handshake was the next concept we talked about. So you have these four core histones. They have three alpha helical regions that form this characteristic structure called as the histone fold. There is alpha 1, alpha 2, and alpha 3, which represent the three alpha helical regions. L1 and L2 are the non-helical loops. Two different histones, they sort of form a handshake between each other, where two different in the octamer, the three helical regions, they are arranged in a manner which gives rise to the histone fold, like we just saw. So H3H4 dimer and the H2H2B dimer of dimers, they interlock to give this structure, and it gives rise to the stable core octamer that we eventually see. Now you can see here the amino acid sequence of the four core histones can be seen, and you can specifically look at the N-terminal tails, which have multiple sites that can undergo many different kinds of modifications. That actually controls all of the epigenetic variation that we talked about, or it controls a large part of the epigenetic variations that we talk about whenever we refer to any post-translational modifications within the cell. Um, so Yeah, so you can see there is different numbering. I'm not, again, not going into too much detail, but you can see there's a different numbering. You can see that the length of the histone tails are also different. H3 actually has the longest tail. It's, I think, a common question also in some exams and all. So you can refer to that as well. And you can see that largely there are some common residues on which these modifications take place. Mostly it is lysine and arginine. Uh, most commonly, and they have these marks that we already have talked about. Um, the types of modifications can vary. These include acetylations, um, acetylation, phosphorylation, methylation, ADP ribosylation, and sometimes ubiquitination and sumylation. We talked about DNA methylation also. So this is something very distinct from histone methylation. Histone and DNA methylation have two completely different purposes. Please make sure you do not mix up the two of them. We have already discussed this in detail previously, so just revisit that class to refresh your memory. But yeah, what we saw here is that between your between these two forms of acetylated, non-acetylated, and methylated, non-methylated histones, there are certain enzymes which come in, HATs and HDACs, which are responsible for the transfer of an acetyl group and the removal of an acetyl group, respectively, or with the transfer of a methyl group and removal of the same, are very crucial enzymes in this context. Apart from that, we talked about histone variants, which are usually found to be present in certain contexts, especially during certain stages of the cell cycle. Um, these variant genes are more important in those states only, like macro H2A, which we talked about histone H3.1, 3.2, 3.3, H2AZ, which all take over the process of chromatin remodeling under some very specific contexts. Then we talked about histone modifications in more detail. We talked about the difference between epigenetic code or the histone code, which is again a bit different from what we have learned about the genetic code, but then it also has its own um, attributes. Histone post-translational modifications, they also have a different message to convey, and therefore the histone code is also something that is quite well understood. So largely this code is maintained by certain types of enzymes called as writers, readers, or erasers, three classes largely. So again, you can learn about all the different post-translation modifications that can exist in this box. You can look at the interchangeability between these two processes from HATs and HDACs and the different enzymes which actually comprise these classes. Just a second. Yeah. Then we talked about different mechanisms that underlie the effect of histone post-translational modifications, recruitment of histone modifiers. Um, like there can be histone acetylation, the effect of it, the different ways in which histone code has a role to play, X chromosome inactivation in cell mitosis, transcriptional inhibition, and transcriptional activation are actually the major classes that are very, very, very extensively studied. That what is the histone mark you observe at a particular region can dictate a lot. So that's something very crucial. You have DNA damage repair, you have cell differentiation, etc. Then we talked about histone lysine proteinylation and evolutionarily conserved post-translational modification. Um, HTK4, HTK4ME1, HTK4ME2, ME3, all three of them. You can see that depending on the ChIP-seq read, you can see that HTK4, each of these marks has a different attribute associated to it. Again, you can read about that, or you can revisit that uh, to brush up the basics, but each of them have a different role to play. The enrichment of each of these PTMs or post-translational modifications is quite different. For example, the HTK4ME3 is enriched in promoters of active genes, and it helps recruit the transcriptional machinery and basically is an active mark, which is surprising considering that methylation is usually considered to be repressive in nature. So again, that brings us back to the fact that it's not as transparent as we might think, and there are subtleties which need to be addressed. So this is the sort of an um, overview of what the histone octamer interacting with DNA kind of looks like. There are different omic techniques that can be used to study the genomic localization of histone modifications. ChIP-seq, cut and run. We have discussed a few of those techniques: cut and tag, chill-seq, dimelo-seq, etc. are all the different methods. Then, after a recap of the NGS methods, again, I'm not going to visit that. Uh, just, yeah. Then we looked at the overall strategy for data analysis. So you have hypothesis designing, experimental plan, NGS outputs, data preprocessing, alignment, and downstream analysis. So some key definitions were initially discussed. Then we understood different data formats that includes FASTA, FASTQ. FASTA is the raw data file you will get, and then it can be used. FASTQ can be used for storing data, getting your quality scores, and basically pre-processing your data properly. Then there is something called as the Fred score, which represents the probability that the corresponding base call is incorrect. And this is how the Fred score is calculated. Again, we have discussed that also. Then we talked about other data formats like SAM, BAM, GFF, GTF. SAM is the alignment file, and it's a very heavy file, so it can often be converted into the binary version known as the BAM file. Then you have GFF and GTF files, which are for general feature format, gene transfer format. These are also extremely important. You have your bed file, which represents the information or the features that you found in the molecule. Then you have GFF, GTF bed formats also. You have VCF or the variant call format that contains information about genetic variants found at specific positions in the reference genome. Next are the parameters which are taken care of while pre-processing of data. I think I'll end the discussion today and overall summarize, because the rest of it I think we've very recently revisited as well. Um, Let me switch on my video. Yeah. So, like I was saying that for fast QC, which is the quality control or quality check of this um, entire data analysis protocol. For fast QC, what you basically want to look at are the fact that whatever data you're using for further analysis, whether or not it even meets the basic criteria that it should meet, so that any results you get are not inaccurate, are not unreliable, and the computational energy and of course your own physical energy that you're investing in it does not like completely go to a waste. So there are a few parameters which are checked and which are approved by the FastQC tool. So that includes, first of all, basic stats, of course. Then you have per base sequence quality. This is how it looks like, the per base sequence quality. Preferably, you should have everything within the green zone. This is quite poor data and not something you should definitely look at. If your box and whisker plot is too wide, that's also not very advisable. And a very good read would be something which lies largely in the clean zone. Then yeah. Um, then you have the per tile sequence quality, which only appears if you're using an Illumina library. So it gives you that within that particular tile, how good is your read quality for that entire tile. That if there was a flawed tile, for example, within which all of the reads are not properly sequenced, then you would have to accordingly adjust your parameters. Then come per sequence quality scores, which illustrates the total number of reads versus mean sequence quality score, and allows us to see if a subset has universally poor quality. Um, Per base sequence content also comes in. So how diverse is the sequence of that particular how diverse is the base composition of that particular sequence that you are chasing? That also makes a lot of difference. Just excuse me a moment. Yeah. Uh, okay. So, sorry, I got a call. So, if basically this idea of this particular parameter is to check that there is no there's no bias in your sequences. There are no overrepresented sequences. Your adapters are removed, and like for example, if you take 10 reads and you sort of align all 10 reads and you see that there is a preference of certain bases at certain positions in all of those reads, then that is in sort of incorrect, and it's a contaminated library, and it's not something you should proceed with further, unless it's an adapter or it's a primer or some sort of that sort of a sequence, which should automatically get removed by the end of your fast QC cleanup. Uh, so that should work out fine, but it's a good idea to trim sequences at least till that level. For example, up to here, it's a good idea to probably trim your sequences because there might be some bias which is there, or maybe the initial sequencing might not have proceeded properly. It's normal and expected, but trimming should be done properly. Then you get your per sequence GC content, you get your per base and content. GC distribution should be more or less sort of normal everywhere. Per base n content, which shows that no base call, if present, it should be also not very high. And in most cases, a low proportion of n's appears towards the end of a sequence, which is also fine. Then you have sequence length distribution. So the in terms of short rigged sequencing fragments of uniform length, um, that should ideally be generated, but that can also change over time. Then you have sequence duplication levels. Um, when we talk about sequence duplication, usually two sources are attributed to it. One is PCR duplication, and another is that yeah, it's PCR duplication usually is what happens, and they misrepresent the total true proportion of sequences in your starting material. However, duplication is normal in case of RNA-seq for abundant reads, and that should ideally be removed. I'll complete Trimomatic and then we can pause today's class, and in fact the course for that matter. So Trimomatic is the tool which is used for chopping up these regions of your sequence or of your reads. So it's a multi-thread command line tool which is used to trim and crop Illumina data and remove adapters. Two major modes are paired end and single end mode. Trimomatic works with FASTQ files. There are two major modes of the program, like I mentioned, paired end and single end as well. And the sliding window approach is what is usually taken. There are a number of commands which need to be followed whenever you are running Trimomatic, Illumina clip, sliding window, max info, leading, trailing, how many bases need to be cut, whether it's from the leading end or the trailing end, what is the minimum length of the read that should be maintained, should we at the time of trimming follow a certain parameter, that if a certain score or if a certain threshold is not met, then that region should be trimmed off, or whatever parameters are necessary. All of that should be mentioned and uh, would be followed by the command line. Then we understood each of these parameters in detail. Illumina clip, which basically sort of removes adapter or contaminant sequences. Um, when the end of the fragment is reached, sometimes the sequencer continues to read through into the adapter, so that all those sequences should be completely gotten rid of. And yeah, Trimmatic includes another adapter identification strategy, which is called as the palindrome mode. So you can look at that as well. So the palindrome approach basically checks for two overlapping stretches which have the common contaminant, and then it gets rid of that. Uh, then it uses a seed, which is for perfect or sufficiently close match. And then it gives us a score, an alignment score. And it takes a sliding window. It slides a window if it calculates the average quality. And if the quality score does not meet a certain threshold, then it trims right at that position. So that's pretty much how it works. Then you get minimal read length, additional read length, error sensitivity, etc. Leading, trailing, of course, means from the end at which the cut or the trim should be made. Crop, head crop, min length, top thread, and top thread 64 course, we have discussed all of this. This was a example of how chemomatic works. And then we had to introduce the reference genome. We understood how alignment works. And yeah, this was again, and now I'm going to brush through it just so that we can wrap up today's class. So this is the complete human genome sequence, the labs that contribute to the reference genome, the concept of indexing, why it's important, two strategies, global and local alignment. Then we discussed the alignment tool STAR. Very recently, I've revised this as well. Splice transcripts alignment tool reference, usually used for RNA-seq data analysis, searches for a seed, and then it aligns the sequence properly. Then we talked about Botai as well, or Botai 2, which is also used, the comparison between SAM and BAM tools, SAM and BAM formats for data files. Then we talked about DNA protein interactions. This was relatively much recent. Talked about initially some the way in which DNA is able to access protein is able to access DNA via the major group, different types of DNA binding proteins, helix turn, helix proteins, helix loop, helix proteins, the leucine zipper motif, beta sheet recognition motifs, zinc finger motifs, transcriptional regulators, and the way in which they bind, the way in which they are able to access DNA, the occupancy associated graphs as well, or curves as well. Then the way in which transcriptional regulators are able to bind, how they're able to open up chromatin. We discussed that. Then we discussed in vitro DNA protein interaction studies from footprinting assay onwards. And this was, I think, really recent. So yeah, I think that's pretty much. And I think lectures have also been uploaded till this point. So I will stop um, my side of the story for now. What I do want to say, um, however, as um, sort of a conclusion of this entire course, my objective with this course was A, to teach, B, to learn myself, because I'm also an active researcher at this point. I have had the immense privilege of receiving this fellowship. And under the purview of that, A, I get to teach, I get to better my own basics, and I get to learn new ways of explaining things. I know there's a lot of scope for improvement, and I hope to do really well. Um, but along this journey, I've also learned along with the people who have participated. It's very unfortunate that the last two lectures are, I couldn't take, like I am uploading these lectures for easy understanding. And I want it to be accessible to everybody as well. And thank you for being a part of this course. Thank you for uh, bringing up really nice questions. There were a few students who were very enthusiastic, got me really nice questions, were active over email. And I hope it helps you in some part of your life. I hope you got to learn something interesting, because I did, and I enjoyed it a lot. I know it was a very long course. It was, I think we've been attached to this course for around 6 months or so. That's a really long time. So, thank you for being a part. This is going to be the last video. All of it will be uploaded over YouTube. All the associated material is going to be shared over email. Everything is complete, but I am always available if there's anything you wish to talk to me, anything you wish to learn from me. I'm always available over email. Um, and yeah, that is it. Thank you very much, and good night. Goodbye.